Here's the latest audio research news from the transmitter. Unprecedented dorsal root ganglion atlas captures 22 types of human sensory neurons by Callie McMurray. A new atlas of cells in the dorsal root ganglion details several novel types of sensory neurons and provides molecular and cellular targets for potential pain treatments. The atlas appeared as a preprint on bioRxiv last month. The data are also freely available online. The dorsal root ganglia dangle off the spinal cord and house the cell bodies of the sensory neurons that innervate the entire body. It's a great tissue to study to come up with new targets to treat chronic pain, as well as to understand how the nervous system processes touch, temperature, and other somatosensations, says Theodore Price, one of the investigators on the project and professor of neuroscience at the University of Texas at Dallas. But by virtue of their location, dorsal root ganglia are difficult to obtain. Researchers can't order the samples they need and must instead rely on donations of varying quality collected in different ways from an array of people. There's a lot of uncertainty, Price says. To assemble the new atlas, a team consisting of Price's lab and 3 others collected dorsal root ganglia over the course of 2 years during surgeries, autopsies, and postmortem organ donations. The result contains more than 53,000 neurons and nearly 500,000 non-neuronal cells from 126 people spanning infancy to age 90. This is a real landmark study that really goes above and beyond in just sheer scale and number of samples to really get a very comprehensive view of what's going on in the human sensory neurons, says Diana Batista, professor of cell and developmental biology at the University of California, Berkeley, who was not involved in the project. This is unprecedented. The team identified 22 types of sensory neurons and 10 non-neuronal cell types based on the RNA they express. The sensory neurons include 10 not previously captured in humans, involved in sensing pain, heat, touch, and body position. Finding new sensory neuron types for sensory biologists is not unlike finding new chemical elements for chemists, says Alan Herman-Poole, assistant professor of neuroscience and anesthesiology and pain management at the University of Texas Southwestern Medical Center, who was not involved in the effort. It's truly fundamental, and it's very important. The methods each lab used to sequence the cells varied in the percentage of cells captured and sequencing depth, yet each landed on the same number of cell types. This consistency among the teams, as well as with previous human and mouse atlases, speaks to the fact that they were able to have some sort of robustness in this analysis, despite having to pull together a lot of very disparate data sets, says Zach Lewis, senior scientist at the Allen Institute for Brain Science, who was not involved in the project. It's painting a pretty solid picture. Researchers can use the atlas to probe if we really understand how the somatosensory system in humans is organized, Batista says, such as whether humans have the same cell types as mice and other model systems. The resource also illuminates cells and molecules implicated in pain processing in humans, which can be contrasted with those in mice, both to identify rational targets for new pain therapies and to explain why past therapies failed to translate, Poole says. The diversity of the donors makes it possible to ask questions about sex and age-related differences, too. The proportion of most neuron subtypes is similar between men and women and across age, the Atlas team reported in their preprint. The more donors you have, the more features you can start to look at, says project investigator William Renthal, associate professor of neurology at Harvard University. The sheer number of donors in this atlas, nearly 4 times as many as in previous ones, gives us a lot more confidence in where there are similarities and where there are differences. In future studies, Renthal says he hopes to examine how the medical histories of the donors, particularly those with pain disorders, correlate with gene expression in dorsal root ganglia. The team also attempted to harmonize the different naming systems for sensory neurons. The literature is pretty messy on this front, Renthal says. Some researchers name cells based on their function, for example, a low-threshold mechanoreceptor, conductance speed, slow, unmyelinated C versus fast, myelinated F fibers, or peptide release, peptidergic nociceptors. But in the body, cells don't have names, Renthal says. Each cell is an individual. They all do their own thing. The atlas introduces a more detailed naming system that contains the fiber type, key genes that the neuron expresses, and functional information, for example, A proper. HAPLN4 is an A-fiber proprioceptor neuron that expresses the HAPLN4 gene. Eventually, we can all be speaking the same language, not just about human DRG cell types, but comparing across different species too, Lewis says. That's a nice effort and really does help to advance the field. This audio was created using artificial intelligence. Stay up to date with the latest neuroscience news and perspectives at thetransmitter.org. Follow Audio Research News on Spotify, YouTube, or wherever you get your podcasts.